Morning. So today we are starting with sex determination in humans. Now, when we are talking about humans, we know that oocyte has a single X chromosome. Just a second. Okay, so the oocyte has a single X chromosome, and the sperm has, so it has the X chromosome, and the sperm has an X chromosome or a Y chromosome. And when we are drawing these gametes in the checkerboard, we see that the XY combination always gives us a male. So it is evident that since we have done Drosophila earlier, where we understood that the ratio between X to A becomes important, but in case of humans, the Y chromosome is a deciding factor to decide that whether the embryo will be a male or a female. Now, certain events which are important with respect to the development of the human embryo. So if we look at the genetic material XY male, and XX is female, when we are talking about the gonadal sex, undifferentiated structures begin to develop as testes or ovaries. That happens at 6 weeks after fertilization. If we look at the phenotypic sex, which happens, look at the timing, it happens at 8 weeks after fertilization. This is where we get the development of the external and the internal reproductive structures. And this development takes place as a male or a female in response to the hormones that are being produced. The concept of gender identity, that is strong feelings of being male or female, that occurs during childhood or possibly earlier. And finally, sexual orientation, that is attraction to the opposite sex, that happens again from childhood. So these are some of the important points regarding sexual determination in male and female. We move on to the next slide. Now, when we are talking about the chromosome, as you can see, this is the X chromosome. And this is the Y chromosome. The sex with two different sex chromosomes, that is a male. If you look into the male, and if you look at the sex chromosome, one is X and the other is Y. That is a heterochromatic sex. When we are looking into the female, where the female has both the X chromosome, that is a homogametic sex. In males, so as I said just now, the concept of heterogametic and homogametic. In humans, males are heterogametic, that is they have XY, and females are homogametic, that is they have XX. So that is the normal way in which sex is determined in humans, where Y chromosome is playing an important role in determining the sex of the embryo. How does the Y chromosome play a role? We'll get into that in the next slides. But before that, there's another important information. In birds and snakes, for example, males are homogametic and females are heterogametic. So that is the ZZ homogametic and ZW heterogametic mode of sex determination in birds and snakes. In case of humans, the X chromosome has more than 1500 genes, and is, as you can see, if you look into the picture, I showed you the picture at the beginning, you can see the size of the X chromosome and the size of the Y chromosome. And the X chromosome is much larger than the Y chromosome, and the number of genes present in the Y chromosome is only 231 protein coding genes. In meiosis, in case of males, the X and the Y chromosome act as if they're a pair of homologs. So these are some more important information regarding the sex chromosome in humans. And now we move on to the next slide. Now let us see how Y plays an important role in determining the sex of an embryo. Let's start with the females, because females are homogametic and they have two X chromosomes. So when there is no Y chromosome, as you can see, that if there is no Y chromosome, then there is this, the Y chromosome is capable of producing, the Y chromosome has a gene, which is known as Sry gene. And the Sry encodes a protein that causes the undifferentiated gonadal tissues of the embryo to form testes. Since there is no Y chromosome in females, and this protein that is encoded by the Sry gene is known as testis determining factor, TDF. So again, once more, there is this Sry gene present in males, which encodes a protein that facilitates the undifferentiated gonadal tissue of the embryo to form testes. Now we come back to the scenario of ours. In females, there is no Y chromosome. So in the absence of the Y chromosome, the Sry gene and the TDF, so no TDF is produced, no testis determining factor is produced. So in absence of the testis determining factor, the cortex of the embryonic gonads, so the embryonic gonads develop into ovaries. And since there is no Y chromosome, no testis development took place, no testosterone production. So in the absence of testosterone, the embryo develops the female characteristics. If, on the contrary, we look into the heterochromatic sex of the male has a Y chromosome, as you can see, and there is this Sry gene. As I said, the Sry gene produces this protein known as the testis determining factor. So in presence of the testis determining factor, what happens? The undifferentiated gonadal tissues of the embryo is modulated to form the testes. So the TDF, as you can see, so this is the XY chromosome. We have that small Sry gene, which is producing the testis determining factor. The testis determining factor, as we have realized, is produced from the Sry gene present on the Y chromosome. The testis determining factor induces the medulla. So this is the medullary part. The medulla of the embryonic gonads to develop into the testes. Remember, in this case of the females, just a second. In case of females, the cortex developed into the ovum. But in case of males, under the influence of the testis determining factor, the medulla is developing into the embryonic gonads. The testis determining factor induces the medulla to develop into the embryonic gonads, finally the testes. And once the testes are formed, they produce this testosterone hormone that initiates the development of male sexual characteristics. Now the figure at the extreme right is that in the early human embryo, as you can see, there is this red duct and there is this blue duct. The red duct is known as the Mullerian duct. Remember, the Mullerian duct in the later stages, if it is a female embryo, persists, it stays. While the blue duct is the Wolffian duct. If it is a male embryo, then the Wolffian duct persists. In either or case, if it is a female, the Mullerian duct stays, but the Wolffian duct degenerates. Similarly, if in case of the that when there are these two ducts in the early embryo, so one persists and the other degenerates. So as you can see, in case of females, when there is no Y chromosome and there is no Sry gene, so in the absence of the Sry gene, what we are seeing is that the Mullerian duct is persisting. You can see the Mullerian duct is persisting. The blue colored duct, that the Wolffian duct, has become degenerated, and the Mullerian duct then goes on further to develop into the different female reproductive structures. But in case of males, if I look at the SRY gene, the SRY gene is present. And once the SRY gene is present, the Mullerian duct becomes degenerated. I will come to this concept later on when I'll be discussing in detail the Y chromosome, that testis determining factor targets one gene known as the Mullerian inhibiting substance. This is an autosomal gene. So that the developing cells of the testes secretes this Mullerian inhibiting substance. And as you can understand by the name, since this Mullerian inhibiting substance is secreted, this MIS protein causes the regression of the Mullerian duct in males. So you can see that the Mullerian duct has become degenerated in males under the influence of the Mullerian inhibiting substance, and the Wolffian duct persists, and we finally get the development of the male reproductive structures. So this was a comparison of the anatomical structuring that happens when an embryo that is born with both the ducts finally enters into a path of either a female development or a male development. Now let us come to the Y chromosome. So when we are looking at the Y chromosome, present on the both ends of the Y chromosome are pseudo-autosomal regions. So we have part 2, part 1. So these are pseudo-autosomal regions that have been seen to show homology with the regions on the X chromosome and synapse with the X chromosome during meiosis. Now this pairing is very important. The pairing via the parts is very important for the segregation of the X and the Y chromosome during gametogenesis. So this is an essential phenomenon. Now, if you look at the remaining of the structure, at the extreme end, we are getting the pseudo-autosomal region. Apart from it, the whole of the remaining part, that is the 95% of the X chromosome, Y chromosome, does not recombine with the X chromosome. And hence it is coined as the non-combining region of the Y, or currently it is known as the male specific region of the Y, that is MSY. Now, when we look into the structure of the Y chromosome, so we have understood that at the two extreme ends, we have the pseudo-autosomal region, and the majority of the Y chromosome is the male specific non-recombining region of the Y. And so that those are the part, and this is the male specific non-recombining region of the Y. And as I was saying you that the Y chromosome has two pseudo-autosomal region, as a large central area of MSY that comprises about 95% of the chromosome that is not capable of pairing with the X chromosome that is specific to Y chromosome and has only 22 genes. Now, if we further look into the details of the Y chromosome, let us concentrate on MSY, that is the male specific region of the Y chromosome. As you can see, the male specific region of the Y chromosome is equally divided into a euchromatin portion, the blue color, and a heterochromatin portion. And you people are I think so by now you have this basic idea that the euchromatin portion is the portion that comprises the functional genes, and the heterochromatin portion is the portion that lack genes. Now, within the euchromatin region, now let us concentrate here. Within the euchromatin region and adjacent to the pseudo-autosomal region. So this is a pseudo-autosomal region. The SRY gene becomes active in XY embryos. So this is where the SRY gene is sitting. It becomes, as I just now said in the previous slide, that the SRY gene becomes active in XY embryos, and the SRY encodes a protein that causes the undifferentiated gonadal tissues of the embryo to form testes. This protein is known as testes determining factor. So let's get into that. So this is the SRY. Now, as we saw, as I was just now saying, that at 6 to 8 weeks of development, the SRY gene becomes active in XY embryos. And the SRY encodes a protein that causes the undifferentiated tissues of the embryo to form testes. So once we have this testes, production of testosterone. Meanwhile, testosterone receptors have also developed. Testosterone receptor complex is formed and the signaling starts, and we have the male secondary sexual characteristics. Now, regarding another information regarding this testis determining factor is that the testis determining factor is a transcription factor. That means it can directly interact with the DNA regulatory sequences and bring about the upregulation or the downregulation of its target genes. Two autosomal genes are also target of the testis determining factor. One is the human SOX9, which when activated leads to the differentiation of cells that forms the seminiferous tubules that contain male germ cells. So this is one target. The second target that I explained while explaining the earlier slide was the Mullerian inhibiting substance gene. So if this gene is activated, as you can assume, Mullerian inhibiting substance is secreted from the testes, and this MIS protein then causes regression of the cells in the Mullerian duct. Once the Mullerian duct regresses, any further development of female reproductive tract is prevented. So testis determining factor ensures that once it comes out, it ensures that the embryo will develop into a male embryo. So we have come to the end of sex determining in humans. Just a second. We have discussed this, and this is just another recapitulation of what we have done till now, even though some mutations are also being shown. But let us concentrate on the main images. So if there's this Y chromosome, then what do we have? With the presence of the Y chromosome, there is this SRY gene, which is activated at the 6 to 8 week of development, and the SRY gene encodes for the testis determining factor, which results in the development of the undifferentiated gonadal tissue into the testes. So the moment testes formation starts, as we just now discussed, testis determining factor is also capable of activating an autosomal gene, the Mullerian inhibiting substance gene, which produces the MIS. So once MIS is produced, we see the degeneration of the female reproductive structures. So degeneration of the female reproductive structures takes place. Testosterone is secreted. Testosterone helps in the development of the various internal structures, as you can see: epididymis, ductus differentia, seminal vesicle and ejaculatory duct. Testosterone is further converted into dihydrotestosterone, which develops and helps into the development of various external male structures. Now, these are some mutations that have been shown that if the pathways, for example, if from cholesterol, if the testosterone converting pathway is defective, then this disease occurs, congenital adrenal hyperplasia. If the receptors for testosterone are not properly synthesized, then this condition develops, androgen insensitivity syndrome. If the pathway for conversion of testosterone to dihydrotestosterone is malfunctioning, then this symptom develops the 5-alpha reductase deficiency. And again, if there are malfunctioning in the receptors, then it is androgen insensitivity syndrome. And finally, coming to, this is a picture which I've taken from Sklatzy et al. In 2003. Now, this is a picture that is showing us the MSY region in an elongated manner. And if you can see the male specific region of the human Y chromosome, so there is this X transposed region. It comprises of 15% of the MSY, and it has been seen that in evolution, it has been derived from the X chromosome, and hence it can pair with the X chromosome. The X transposed region is 99% identical to the X long arm 21 region. Next, the second area of MSY is designated as the X degenerative region. So the X degenerative region, the yellow colors, look at the yellow colors. So this comprises about 20% of the MSY, and this contains DNA sequences that are very distantly related to the X chromosome. It has been seen that in the X degenerate region, 27 single copy genes and a number of pseudogenes are present. Finally, the third area is known as the Ampliconic region, which is this. This is the heterochromatic region. You can see the heterochromatic region shown here, and this violet bluish color is the Ampliconic region. It contains about 30% of the MSY, and it includes those genes which are closely associated with the development of the testes. These genes lack counterparts on the X chromosome, and their expression is limited to the testes. So the Ampliconic region is very much specific to the MSY region of the Y chromosome. In this Ampliconic region, there are 60 transcription units that has been divided among 9 gene families. So this much information is enough regarding MSY. And with this, we come to the end of our sex determination in humans. We'll start dosage compensation the next day.